Um, Hello everyone, my name is Elias Ciudad. I am a rising junior in Summer University studying bioinformatics, and this is my project, single-cell RNA analysis of spleen in myelofibrosis patients. And by the way, that's a spleen. I'm going to give a brief overview about the background that I use, the tools, the methods that I use, and also about my results. So I use single-cell RNA sequencing, and I like explaining this using an analogy of a berry smoothie. Say that you want to make a smoothie with different berries like strawberries and blueberries, but you don't care about which one you are using. In that case, you will be doing bulk RNA sequencing. This is because all of your cells in bulk RNA sequencing are going to be mixed, and you're going to get this raw pool of gene expression. However, if you do care about seeing how many blueberries you use, how many strawberries you use, and comparing between them, you're going to want to use single-cell RNA sequencing. This is because if you because you're going to be able to obtain single-cell data on a single-cell basis, and you're going to be able to cluster the cells based off their biological features. And this allows us to study heterogeneity and lineage across different cell types. I also use the Ciudad, the R programming language, which is very well known for its data transformation and visualization capabilities. And most specifically, I use the Ciudad toolkit, and I don't mean a French artist. This was developed by the Ciudad lab, and it allows us to do some pretty interesting omics data visualization, quality control, downstream analysis, and single-cell data integration, especially with Ciudad B5. So going on and to explain what myelofibrosis is, this is a very rare bone marrow blood cancer that is mostly linked to JAK2 and CALR driver gene mutations. What's interesting about this disease is that your hematopoietic stem cells, which are able to differentiate into all the different blood types, cells types in the blood, are going to migrate from the bone marrow to the spleen. This means that you're going to get all these HSPC cells going there. They're going to replicate over and over again. Your spleen is going to get enlarged, and you're going to get cancer. So as we may know before, these samples we obtain for all different places, different patients, different times. They're all sequenced in different machines. So we're going to see some batch effect, which basically means that we are not seeing cells being clustered based on their biological features, but more so by random noise. As you can see on this HuBMAP on the left, they aren't very evenly distributed. So we decided to integrate based off their original identity. And here on the right, we can see that everything is much more evenly distributed. And this will allow us to get more accurate conclusions moving forward. Here are some very broad annotations that I made for all the different cell types. Here we can see that our HSPCs seem to have very sparse reads. So let's go on and see why. This is because, as we expected, we are only going to see HSPCs on myelofibrosis patients. On a healthy individual, you will not see any of these HSPCs that come from the bone marrow. Since they come from the bone marrow, I decided to use a bone marrow reference using the Azimuth software. And it was also made by the Ciudad lab, and it allows us to really like speed up the process of annotating cells and to automate it. I did all of my annotations manually, and it was very tedious, mostly staring at a heat map for hours. But Azimuth is a very reliable reference, and I will definitely use it in the future. And this is how I fared against the machine. As you can see, there's much more of a difference here on the B cells, CD4 cells, your T cells, your NK cells, but they are around the same in the HSPC area, and we're going to be focusing more on this. And these are all the markers that I use to annotate these cells. We can see that there are very different and multiple types of HSPCs, like erythrocytes, GMPs, program Ks, HSCs, and much more. So again, we are very interested in finding similarities and differences between the patients that we had. So we decided to look at the proportion of these cell types per sample, look at the variance. We were able to identify that the most variable cell types are actually late erythrocytes, HSCs, and GMPs. But these are just numbers. We want to see how does this even relate to clinical data. So the clinical data gives us information of the variant allele frequency of all these 7 patients, 3 of which are normal, so they're healthy. We decided to focus more on the MF4 and MF5 patients. This is because they are the only patients with CALR mutations. and also happen to have very low JAK2 gene mutations. We want to see if this is mutually exclusive and to see why. Is it maybe chromatin accessibility? Is it a cell type? Is it DNA? Or is it just mere chance? We decided to do some correlation and to subdivide our samples between JAK2 mutants and CALR mutants. Here you can see that across all the cell types, program K had the highest correlation, and it also had the best p-value. So we're going to be focusing on these for the analysis. As we can see here, our CALR mutants seem to have more program Ks than the non-CALR mutants, and this is also ordered in an increase of JAK2 variant allele frequency. We decided to delve deeper, and we can clearly see that these two patients, which have CALR mutations, have a way higher percent of program Ks compared to these other samples. Here we decided to do differential gene expression. We're going to compare each of the green circles to the samples in the blue circles. And then we're going to intersect them based off their upstream genes and their downstream genes. And we did that, and we obtained this beautiful heat map. We're hoping to identify subgroups, and so far we can see that sp29 and sp30 are very closely related. And we are particularly interested in seeing that this gene, the TRHDE-AS1, is completely or almost completely inactive or downregulated in these two samples. So we wanted to see if this is like exclusive of these two samples, if it's exclusive by the program K cell type. So we decided to look at all the program Ks across samples, and we can see that it's not very well conserved. It happens to be the same across all samples. We were hoping to see a subgroup, but we didn't. But maybe we were thinking, maybe if we go by cell type, this gene is going to be more conserved, more uniform. And turns out it wasn't. We weren't fully able to see any transcriptional differences, but we decided, we had this other idea. Wait, if we can see more program Ks in these two samples, maybe the cells are just replicating more. So we decided to look at the cell state, like G1, mitosis, G2. Or before that, we decided to see if there were any, if it was exclusive to cancer patients, and it was not, because SPN11 and NYU and N3 are normal spleen samples, which also had the TRHD-AS1 gene completely downregulated, so that completely discarded our initial hypothesis. And then we went on to the cell types, the cell states of these cells, and we decided to subdivide them into cycling, being the G2 and M phase, and the S phase, and non-cycling for the G1 phase. This is a HuBMAP base of our HSPCs. We tend to see that here are the program Ks, and they seem to have the same amount of G1, G2, and S, but we ran some more tests, and we can see that there are barely any significant differences between these two subgroups on CALR or JAK2 patients. In fact, we, based on proportions, we saw that there were actually more cycling cells in the JAK2 patients than in the CALR patients. That's science. So the conclusions of my 9 weeks here is that Azimut is a very faster and reliable method to annotate cell types, and it's much better than doing it manually. Cell types are variable across samples, especially HSCs, late erythrocytes, and GMPs. The remutations are in fact correlated to a proportion of program Ks, but we don't know exactly why yet. We can also see that there were in fact transcriptional differences between CALR mutants and patients with a high JAK2 variant allele frequency. And in the future, we hope to run the same analysis on different cell types like GMPs, EMPs, ELMPPs, look at other mutations maybe, run more statistical tests of the cells in different cycle stages per sample by cell type, and explore the gene ontology of our differentially expressed genes. And what I learned in this internship is that science is very complex. You can start from DNA, RNA, proteins, gene expression, then you go on to the cell types, And whenever you run an analysis, you want to compare multiple cell types across multiple genes. And it was certainly a learning experience to run these kind of experiments more independently. I also learned how to use multiple toolkits in R, like the Ciudad package, like I mentioned before, and which was also kind of confusing to program. But there's always room for optimization, and going on the internet and asking for questions, asking for advice is always helpful, because one thing that I learned also here is that I am never going to be the first one asking the question. There's always someone out there that can help us. And this also means that science requires a lot of collaboration. And this is what HuBMAP is all about. I'd like to thank everyone in my lab, more especially my mentor, Madeline Kowalski, our PI, Rahul Sotilla, and Maya Van Santen for being a very nice program coordinator, and to the NIH, the HuBMAP organization, and to the New York Genome Center for having me during these 9 weeks. Thank you.